Good afternoon. Thank you for deciding to spend this pleasant sunny afternoon, which is so rare this spring here. Let's go back to some pictures that I showed to you a week ago and 2 weeks ago, and let us remember what we discussed. Specifically, those are things that we discussed to some extent. We discussed territories, and we discussed this slow decay of probability distribution for the contacts that we discussed. But there are many other things which we did not discuss at all. And I will not really discuss many of these things, mostly because I know for sure that some people in the audience know more about this than me, and so there is no point. So I will only briefly mention that many of these things, such as this slow decay of probability distribution at relatively short distances, and specifically these points at studs and corners, all of that is nicely explained by the most beautiful idea of loop extrusion that, again, many people here know more about than I do, and so therefore I am not in a position to tell you anything new about it. So it was basically fruitfully formulated in these papers here, and it explains lots and lots of things. In fact, it was formulated, it was formulated in these papers over 20 years ago, and then over 10 years ago in this paper by Alipur and Marco, but really it started when these papers appeared. So there are lots and lots of things are known about this loop extrusion process. The most beautiful of them is the story of the thing that is called CTCF, and don't ask me how to open up this acronym. But the basic idea is that there are some specific places, specific sequences which stop this active extrusion of loops, and this creates all sorts of beautiful stories, which again I will not talk about. Moreover, in the group of Case Decker, not far from here in Delft, people were able to observe the motor activity of these loop extruders. So you see how beautifully it works. It generates this loop, which you visualize by placing DNA in the flow. And there are beautiful stories about it. Again, there is no point for me to speak about it. I was most surprised by this story, the fact that these condensing motors, they can bypass one another, which is mind-boggling to me, but that's what you see in these pictures. So that's a fact. So there are many beautiful stories about this loop extrusion, and you really should not rely on me to get this, because I am not an expert in this. Instead, I will tell you something else, because my subject, as I formulated, is polymer physics. So I will speak about polymer physics angle of view. And here is a picture of nucleus. And you see that it's not uniform. So far, I was considering this chromatin is uniform media. I told you it was similar to average temperature of all patients in the hospital. So they found density by dividing the number of monomers by volume. So you see that density is vastly different. There is denser part, which is called heterochromatin, and which does not There is almost no transcription there, while in less dense euchromatin there is transcription, and the heterochromatin typically, not always, but typically sits around the periphery of the nucleus and close to these nucleosomes, but there are exceptions and so on. So what would be the nature of this? Of course, in fact, this euchromatin and heterochromatin are not only visible in this type of photographs, microscopic photographs, they are also indicated in some form in high-C maps. Even in the first paper about high-C maps, they made this drawing indicating so-called A and B domains. And A and B domains, there is still subtlety between this and euchromatin, heterochromatin, but basically, to the first approximation, it's the same thing. So there is indication even in high-C maps that there are some separation of domains. So the first inspiration, how you would think what would be this separation between different states, between like different phases in the nucleus, that would be, of course, microphase separation of block copolymers. Of course, speaking about block copolymers, I cannot avoid mentioning this paper by Ludwig Leibler. Interestingly, in the review by Tim Lodge, he says, legendary paper. So it's legendary paper. So legendary paper, I don't think Ludwig predicted all these phases, but this is what present-day block copolymer people claim to have observed. So there are indeed many structures that you can observe even in the block copolymer system, which is much simpler. So that would be the idea how to explain heterochromatin and euchromatin. So what is the explanation, hand-waving explanation of this block copolymer microphase separation? It's very simple, in fact. If you have a red block and blue block, and they, by nature of their solvability, they do not like each other. They want to separate. Well, normally, if they were not connected here, you would have microphase separation. You would have red phase and blue phase, and that would be the end of the story. But now they are separated. So red parts somehow assemble together, but next to the place where red parts assemble, you must have blue parts. So for instance, you can assemble some red pieces, red dots, but then these red dots must be surrounded by the blue sea and vice versa. So that's the zoo of structures that you can obtain. So combination of two things. They don't like each other, and they are connected in the chain. That's it. So both of them potentially may be present in the chromatin, so that would be what people think about. Indeed.
Quick question. Those different structures, they're supposed to arise if you have blocks of regular sizes?
Yeah, well, to tell the truth, I do not know exactly when, what are the conditions of formation of some of these exotic structures. But basically, you can obtain very rich phase diagram even if your block copolymers are completely monodispersed, which means you have all blocks of blue are the same length and all blocks of red are the same. Blue and red are not the same length, but all blues in the system are the same. So immediately, of course, people jumped on this bandwagon, and for instance, this is a paper from the group of Andrew Spakovitz in Stanford University. They simulated some sort of block copolymer model, and sure enough, assuming that there is some maybe epigenetic modification of some of the histones which make them attract more than others, you obtain some sort of phase separation. No surprise there. More sophisticated simulation around the same lines was due to the group of Sam Safran in Israel. And you see, varying the energy of chromatin self-attraction, they can obtain chromatin that sits inside and mostly along the surface or mostly in the middle. And if you change the quantity of chromatin inside, you obtain different distribution of. And this is all very easy to realize, given that this quantity here is the strength of attraction. You assume that some of these monomers attract to the surface, so they wet the surface. And this is pretty easy to understand all this. So in my language, it supports the idea that this is sort of a polymer globule sits inside, and this polymer globule behaves like almost a liquid, and there is a wetting of the surface, so there is no surprise here. You basically obtain from your simulation what you put into your model, in my opinion. Then these people went further, and they observed experimentally that the distribution of various parts, which they label some of them green and some of them red. I don't want to go into details, but you see that the distributions are not uniform, and they said, oh, this is a sign of microphase microphase separation, this type of things. So they considered a model in which a polymer consists of many blocks. You see green blocks and red blocks. In my cartoon, it was blue and red, and this is green and red. That's a big difference, of course. But otherwise, how do you regulate the interaction? Their idea is that you can regulate quality of solvent, regulate interaction between different units in the following way. They imagine that there is, in the solvent, there is some protein complex, which is shown here by these yellow balls. And these yellow balls can attach only to the green blocks of your polymer. And when they attach to the green blocks, they modify interaction of green blocks with green blocks or modify interaction with green with red. And then you obtain a variety, controlling the concentration of these yellow protein complexes. They obtain a variety of phases, of block copolymer phases, pretty much the same as and the D block, maybe not that reached because they did not start to explore the whole richness of this phase diagram, but they obtain reasonably rich phase diagram. And this is the idea. It is quite a significant activity when people try to model segregation, observed segregation between hetero and euchromatin, but by various models of this type. You assume it's a rich field to play games. You assume different types of interactions. You assume, for instance, this adding proteins. You obtain various equilibrium diagrams. I just want to point out that similar ideas existed in the literature many, many years ago, long before anybody was able to draw pictures of this color and beauty. So I call it cave art. But you see that the idea is the same. You have this globule, and you have this open part, and this is microphase separation, and this is controlled by, and there is phase first order phase transition because it's controlled by the number of black particles that are absorbed on the chain. So it's the same idea, much more primitively executed, but the idea is very simple. More radical suggestion along the same lines, the idea is that this segregation between heterochromatin and euchromatin is the equilibrium phenomenon. It's the equilibrium driven by the difference between interactions between different units. More radical idea along these lines is to make the comparison of chromatin folding with protein folding. Of course, chromatin is 1,000 or more times longer polymer chain. But it is still a heteropolymer. So protein folding, for those who don't know, this is my favorite cartoon about it. So you have a chain of units of different units. It's heteropolymer or copolymer, but not block copolymer, but just not random. It's just a copolymer with certain sequence of certain units. And when it folds, it sort of reveals in 3 dimensions the information that is hidden in 1-dimensional sequence. So this idea of translating information from 1-dimensional code to 3-dimensional code, people try to apply the same idea to chromosomes. So one way to think about it is in terms of funnels. I don't like the language of funnels, to tell the truth, but this is just me. I don't like it because if you ask, this looks like a graph to me, a plot of some function, right? So I was told in Russian school when I was a boy that a plot, there should be vertical axis and horizontal axis. But if you ask what is the horizontal axis, there is no answer. Nobody knows. Maybe something incredibly multidimensional, which makes no sense, or some people even dare to say that this is entropy along this axis, but then I remind to you that derivative of energy with respect to entropy is related to temperature, and it must be positive. And the derivative of this is decidedly not positive. So there is no way to explain what that means, but it's sort of appealing picture. So people very actively explore the idea that maybe some sort of folding funnel concept or imagery may help to explain what happens in the chromosomes. Well, if you take it seriously, then you may want to write down energy of such object. And this energy is nothing else but the sum of pairwise energies. So you have monomer S and monomer S prime. P of S is chemical name of monomer S. P of S prime is chemical name of monomer S prime. For proteins, there are 20 different chemical names. For chromatin, many more. And you have to know these potentials, these energies, for all possible pairs of these monomers. And the number of possible pairs for proteins is about 200. And for chromatin, it runs in many hundred. So there is famous statement by John von Neumann that with 5 parameters we can fit an elephant. Or more accurately, I recently read, with 4 parameters I can fit an elephant. And with 5, I can make him wiggle his trunk. So here you have a model with 500 parameters. Obviously, with 4 parameters, you fit an elephant, and with 500 parameters, you can definitely fit elephant's genome or whatever you want. So that, to me, is a dead-end approach, but it's my opinion. And other people may be smarter and may prefer different approach. So this is the state of affairs. People assume that the difference between heterochromatin and euchromatin, between A and B domains, between domains, is governed by energies, simply by energies of interactions. And then people perform various simulations or analytic calculations and go along that line. This is a possible approach. I do not know, maybe it's true. But there is a different approach altogether. Completely different idea. So maybe, just maybe, this is not an equilibrium phenomenon. Maybe it's not governed by purely balance of energy and entropy as these block copolymers or folding proteins and so on. Maybe it's something else. Indeed, we know that there are phase transitions, phase separations in the systems which are not governed by energy gain. They are governed by consumption of energy, by dissipation. Some phenomena like this, recently most popular by Mike Cates and his co-worker, the idea of random tumble particles or active Brownian particles. These objects undergo so-called motility-induced phase separation, here shown for some particular model. Very clearly you have phase separation between more active particles and less active particles. Jean-François and myself, we developed a different idea, which probably in fairness, if you count how many followers are there for this idea and how many followers of this idea, then this should be for on the entire wall, and this should probably fit in this place. But nevertheless, a different idea is to consider a mixture of particles connected to two different thermostats. And they also phase separate. In some philosophical sense, it is a similar idea that more rapidly moving particles separate from less actively moving or more passive particles. So that is also computer simulated picture indicating such separation. So this is an entirely different idea. This is not equilibrium phase separation. This is not something related to the difference in energies. And that's why I give you three statements which I like. One is attributed to Phil Pincus. If you are in equilibrium, you are dead. So non-equilibrium systems are potentially richer and more interesting. Another is paraphrase of Leo Tolstoy, and equilibrium systems are all equilibrium systems are alike. Each non-equilibrium system is out of equilibrium in its own way. It is the first sentence of Anna Karenina novel. There it's about families, and by the way, I disagree with Tolstoy about families, but that's beyond my point today. And final statement is attributed to von Neumann. For some reason, I now realize, as I speak, I realize that somehow von Neumann apparently was concerned with elephants for some reason. I don't know. In any case, I hope that Phil Pincus would not be insulted by being in the company of Tolstoy and von Neumann. In any case, this idea of phase separation in the system of particles can also be applied to polymers. You can imagine for instance, in this paper by Schmreck and Kremer, they applied Jean-François' idea about different temperatures to polymers. Consider mixture of polymers driven by different thermostats. And they do phase separate, as you see here in this simulated picture. They beautifully phase separate. I don't want now to go to details of quantitative representation, but just look at this picture. If difference between, and as in equilibrium system, you don't need much energy difference to phase separate polymers, which is the beauty of block copolymer story. Here too, apparently you don't need much of the difference between activities to separate these monomers. So since chromatin is long polymer, so that makes the hope that, for instance, if you have transcription in certain part of your genome and no transcription in other parts of the genome, maybe that's enough. The fact that there is transcription, there is activity, the energy is spent there, maybe it's enough to phase separate between euchromatin and heterochromatin. That would be a beautiful explanation, in my opinion, but it's not proven right, and it's not proven wrong today. So it's an open question for now.
So here, I guess the two kind of polymers have different temperature, but for one polymer, the temperature is the same all along the chain.
Yes.
So you know what happens if different monomers at different temperature on one chain? Is it like a block copolymer kind of thing, depending on?
Yeah, it's a block copolymer kind of thing. That's right. And, presumably, it will microphase separate. Not much was done along these lines, but there is no doubt that the result will be like this. It will be
more appropriate, I guess, for the euchromatin and heterochromatin.
Probably, yes. Very true. Yes, very true. There are some technical. This phenomenon, I wouldn't say is completely understood. There are some technical details which we keep discussing about it. So I can go into this, but probably if you're interested, I am prepared to go there, but there are subtleties which we did not completely understand, which is why to go to block copolymers, I am not yet ready. Okay, so, but fact of the matter is there are many people who work on so-called active polymers. Active polymers are different varieties. Basically, you take, let's say, active Brownian particles, connect them by bonds, and say, this is my active polymer. So you see, I made a list of such papers. I don't want to go through all of them. So it's just, I don't think I'm not excited about this. So there are possibilities. But I want to tell you a recent paper which makes, I think, an important novel step along these lines. So what these people do is the following. So first of all, I want to tell you that when you are talking about equilibrium, any equilibrium scenario, you know how to proceed. You know you have to write free energy, you have to minimize free energy. All this machinery of equilibrium statistical mechanics is at your service. You know exactly what you should do. When you are dealing with a non-equilibrium system, you don't have any of this. The only thing that you can do is you can study the dynamics of your system, which is incomparably more difficult task. So in this new paper by Ran Kardar, Arup Chakraborty, and their junior collaborators, just published a year ago or so, what they did is they study the dynamics of such a polymer. And this is the simplest dynamical model, or Rouse model. So I will speak, I chose maybe not the most pedagogical way to go because I plan to go more about Rao's model, but here I just tell you that this is the simplest dynamical model in which friction of every monomer undergoes friction against immobile background. And this part is just the Hooke and spring connection between neighboring monomers. This is what this part is. And most important part is that this is a Langevin equation. There are random forces on each monomer, and each monomer S. At time t, there is a random force. Okay, so it's a Langevin equation, and it causes system to diffuse. It describes diffusion. Diffusion of particles that are connected by the polymer. So this, to make it diffuse, you have to assume that there is this noise, these driving random kicks from the environment. They are not correlated over time. They only correlated at the same time moment. But the novel aspect is that they assume, these people assume that there is potentially correlation between random kicks on different monomers. Monomer S and monomer S prime receive kicks which are correlated with one another. It's in general form written here. They are and this is fundamentally different from what I told you in all these numerous papers. In these papers, I told you every monomer is active particle of its own, which means it's driven by internal noise or external noise, thermostat, something, but every particle is independent of other particles. And these people introduce correlation between different monomers. So this is how they depict, this is their drawing, how they depict it. So they imagine that there is this red, maybe a red piece of your polymer, and there is on this piece enhancer and promoter of the same gene, and somehow they work in concert, and somebody that does something to one of them does also to the other. So there are correlations between what happens on different parts of the same, for instance, domain. And this, by the way, answers your question. This is copolymer this is not different polymers. So it's correlation in one polymer. So we can go back, of course, to uncorrelated monomers. So instead of this general correlation function between monomers, I now consider that there is no crosstalk between different monomers. Different monomers are uncorrelated. Sure enough, they rediscovered the same thing that other people see in this situation. They discovered that, for instance, less active monomers sit on the periphery, pretty much in every model. In model with active Brownian particles, in model with two thermostats, in pretty much any model, if your envelope is stable, its envelope itself is not subject to thermal fluctuations, if it's a rigid object. If it's a rigid object, then less active monomers prefer to sit next to this envelope, which sort of, it's tempting to say, oh, here it is. That's heterochromatin, less active. So that's good. But for that, you don't need this correlation between monomers. So what happens if you do have correlations? If you do have correlations, here is the example which they consider where this function, which sits here and which sort of measures the driving activity at the point S, it is modulated along the sequence S is the coordinate along the sequence, it's modulated. It's every distance lambda, it is above average, below average, above average, below average, and so on. So what then you do? You solve Rao's dynamics equation, and you find that eventually you arrive at the steady state. It does not mean it's protein folding. You do not arrive at a particular conformation that you will publish in protein data bank coordinates of every atom. No, it's not like this. It is fluctuating polymer. It is a Gaussian polymer. They don't introduce any exclude volume interaction, anything like this. It's a Gaussian polymer. It is strongly fluctuating object. Nevertheless, on average, there is time-independent mean square distance between monomer S and monomer S prime at the same time t. I have to drink water, otherwise my voice will betray me.
In this case, what's the structure for CSS prime? The one at the top right of the slide? The coupling between monomers?
Yes, there is S and S prime, two different monomers.
The other one.
The other one, the one at the end of the line.
Here?
No, on the first line. The big, yeah, this one.
Yeah, C of S and S prime is sort of a trivial part. It's related to connections between monomers. I don't know why they write it like this. I would write some constant here. But probably they are smarter than me and they have some further generalization in mind, but I don't see it. I am blind.
Is epsilon supposed to be small in that calculation?
Yes, it's a perturbation theory with respect to epsilon, and you see that this epsilon since here in the first power. So the first term is a trivial Gaussian term, and b square here is the persistence length or Kuhn segment. Okay, so lambda is this length scale. But in addition to this, you have now this oscillating part. And of course, there is decay, but most importantly, there is oscillation, and there is this oscillation with the distance between them. So the end result of it is formulated here, and I wrote it in this red frame. So pairs of active segments are those S where cosine is positive. So active segments tend to move further apart, while passive segments tend to go closer to one another. It is sort of a little bit reminiscent of the idea that they phase separate, but remember, there is no interaction between them. It's purely based on the fact that they are connected in the chain. There is no other interaction between them. So it's a different phenomenon, but I think it's a very interesting phenomenon. And to me, this is only the door. This paper, which I reviewed in some detail, is only a door into a large, potential large space of possibilities to explore different correlated forms of activity between different monomers. I think this is an important step. Now they also make the following interesting observation. Notice that this is a Gaussian polymer. Whatever you do, all this steady state is just Gaussian polymer. Gaussian polymer peculiar, but it's still all Gaussian distributions. And I told you in one of my previous lectures that there is this Gaussian model, which I call spooky action at the distance. When you connect, I probably should have copied it here, but when you connect by harmonic springs, not only neighboring monomers on the chain, but you also introduce harmonic connections between essentially all other pairs of monomers. So these people here showed exactly, without any approximation whatsoever, that any steady-state solution that you obtain by solving the Langevin equation or Rao's model equation, any such steady-state solution is exactly mapped on a particular equilibrium Gaussian model with this distribution of properly chosen distribution of springs. So therefore, if you only observe static, your methods allow you to see only static information. For instance, if you run high C experiments in nothing else, or you done fish microscopy in nothing else, then as a matter of principle, you cannot distinguish the system that was governed in equilibrium by the harmonic springs or by this peculiar correlated activity. So you must have some more information to distinguish between these two, for instance, some thermodynamic information or something else. Yes, Vittorio?
Why this is not a spooky action at a distance too? Why wouldn't you call this also spooky action at a distance? In a way, these correlations, how are they mediated? Anti-correlation.
Well, yes, if you want, yes, yes, you can call it also spooky action at a distance. But remember, it was my way to call it. It's not that authors of that paper with harmonic springs called it spooky action. It's my impolite way to call it. So it's, but yes, you're right. There is also action in the distance because these correlations must have some mechanism. You're right. Yes.
Did I understand it correctly that there are also anti-correlation in this model?
Yes, potentially, yes. When cosine is negative, you can call it anti-correlation, yes.
Because the model, maybe is an opinion, but it looks a bit similar to the Z model when you mediate over, you make this change. Not quite, because in Z model you introduce correlations that propagate through the solvent in three-dimensional space. You introduce correlation between two monomers which depends on how far they are in space and does not depend on how far they are along the sequence. This is exactly the opposite. It does not depend on the driving force of this A does not depend on where this monomer lives in space. It depends only on identity of this. It's like, I don't know why this analogy came to me at this moment. It's like when I I usually work in New York, but sometimes I go see Jean-François in France, in Paris, and it's like I am treated in exactly the same way in both places because it's me, it's my identity. Unlike the other situation in which my treatment does not depend on who I am, only depends on where I am. I don't know if this analogy explains anything, but just came to me on the spot. Yeah, because when one solves the Z model, often you make this change of variable to diagonalize the
solution.
Yes, but it depends on the distance between them.
I totally agree. Yeah, but you make this change of variable. Like instead you assume that is the same. On the other hand, here, the way I see that is very different is that you have negative correlation that I wouldn't see it happening with the aerodynamics.
Well, from that point of view, if you want to have a model in which you have both positive and negative interactions with 1 over r potential, then think about polyampholite, a molecule with both positive and negative charges, and then positive and positive repel each other, negative and negative repel each other, but positive and negative attract each other, and there are frustrations. So if anything, that would be a closer analogy. I think you
It's just that when you say positive and negative correlation, here what's happening is it's not like an anti-correlation between active and inactive segment. It's rather that you have some segment that have stronger thermal or external force and other weaker.
yes.
And so those who have weaker tends to, I guess, need less space to flip around, so they get most of this space.
Yeah, I couldn't say it better than you did.
And just to go back to the CSS prime on the right-hand side, so the different monomers are, there is a correlation between the force on different monomers, or is this one a delta function, the CSS prime?
This is not a delta function, but this connects neighboring monomer.
Only neighbors.
Okay, so as promised, I think I have about 10 minutes to go, right? Is that correct? So as promised, I wanted to tell you a little more about Rao's model in general. In my starting point to talk about Rao's model in the context of chromatin is this experimental paper here. Over about 15 years ago now. And what these people did is they observed, and there are, by the way, quite a few other papers with similar content. For some reason, I like this one. So they observe motion of a particular locus in chromosome, which in this case was telomere, which is the end part of the chromosome. And here is mean square displacement of this locus. Mean square displacement divided by time. The idea is that if it was a normal diffusion, mean square displacement would be proportional to time, and this thing would be constant, but you see that it's markedly not a constant, and it drops, They interpret it, that's similar to Rao's model. So let me tell you a little more, but in order to tell you a little more, I want to tell you that there is something mysterious in this graph which I don't understand. So in Rao's model, they claim that, okay, according to the textbook and according to the story that I will tell you on the next slide, if you observe a Rao's polymer for a sufficiently long time and look at one monomer, for instance, at the chain end, you look at one monomer, at sufficiently long time, you observe regular normal diffusion because this one monomer eventually moves together with the whole chain, and this is normal diffusion. But in chromatin in cell nucleus, there is no room to move the whole thing anywhere because it sits inside the nucleus. So it is not clear to me why this seems to saturate. Saturation means that mean square displacement is linear in time, it's normal diffusion. So what happens at this crossover time, or better to say, at this crossover distance? This distance corresponds to 0.1 micron. What happens at this distance? I don't know. It's mysterious. In my understanding of this experiment fails at this point. I do not know what happens here. It's unclear to me. But here is my story about Rao's model. And my way to describe to you Rao's model is to do generalized Rao's model. Because we discussed with you that, in fact, most likely these polymers, these chromatin polymers, are not Gaussian. So let's not suppose that they are Gaussian. Defining property of Rao's model is not the fact that polymers are Gaussian, but rather the fact that dynamics of every monomer is controlled by friction against immobile solvent. It's not friction against real water. When you swim in the swimming pool, even if you're a very good swimmer, you still generate some perturbance of water around you. This is a swimming pool in which water doesn't care about the fact that you are swimming in water. Just immobile water, nothing happens. So if this is the case, then you can think, first of all, as I said, I assume that it's not necessarily Gaussian polymers, so the size of the subchain of S monomers was like S to the power nu, and maybe I want to think about nu being 1/3, but in general, this nu can be written as 1/d. D is fractal dimension of the chain. It's not the dimension of the space in which we live, fractal dimension of the chain. It's exactly 1/nu. So if a monomer moves during time t, then it has to shift by this distance. This is our regular description. If it has effective diffusion coefficient, d effective, like this, then it moves certain distance. If it moves this distance x, then how many monomers live in that volume of the size x? It will be x divided by b to the power d. It will be just this s. It's the number of monomers that sit inside this blob. So if you move this distance, you have to pull this blob with you, otherwise how can you move, right? So therefore, not only one monomer moves, but this number of monomers move together. And therefore, the diffusion coefficient, effective diffusion coefficient, one monomer, divided by the number of monomers involved in the motion. Because they all swim in this fantastic swimming pool, and each of them experiences friction independently of all others. So you have this number of monomers, each experiences friction independent of others. That's it. And so if you combine these things together, you discover that this is your answer. So x displacement is proportional to time to this strange power. So if d is 2 is Gaussian polymer, then it is 1/4. It is classical Rao's result that one monomer moves like t to 1/4, as long as this S is smaller than the polymer as a whole. As soon as it becomes a polymer as a whole, you are back to normal diffusion, as I told you in the previous slide. In fact, these people who generalized Rao's model, they showed something else there, that there is the coefficient alpha if this fantastic solvent is not just regular solvent that does not react to you moving through the solvent, but it's also viscoelastic somehow. I do not quite even imagine what that would mean, but they then show that there is another alpha which is smaller than unity. And if D is 3, which is the case supposedly for crumpled globule, if this is the way how we describe chromatin, then this is, instead of 1/4, it is 1/5. It's tiny little difference. So how does it work? Well, this green line is 1/4. and this, this orange line is 1/4 and green line is 1/5. Which one fits better? I would say none, but.
So you mentioned these confinement effects that you could have in the nucleus. They are not taken into account here because if you reach the maximum size, you still involve more and more monomers. So essentially the information propagates along the chain and not in space.
Information, I'm not even sure that information will...
Very far away polymers will know about it.
I'm not sure the information, it's an interesting question, but I'm not sure information will propagate along the chain too.
Do you think it will stop at the size of...
I suspect that if there are rigid walls, stress will not propagate beyond, depending on the wall, yes. But I suspect that stress, something important will happen with stress propagation along the chain in these places. So I would be careful about it. So in any case, that's what it is. So of course, I told you about all these other models for rings. And you can ask me, oh, if you take the idea of rings seriously, if you assume that these territories are indeed similar, chromosome territories are similar to territories in concentrated system of rings, can you compute the subdiffusion for rings? Yes, we can. So generalized Rao's model that I showed to you previously with d equals 3 will give you 0.2 for subdiffusion. Double-folded trees model that Jan Smrek and myself developed would give you much lower subdiffusion, t to the power 0.13. Michael Rubinstein and company, loopy globule will give you t to the 0.014. And you see, this is a big fight between these two. Given this state of affairs in experiment, I'm not particularly excited about the difference between these two, but there is difference. And by the way, I think that Michael's work is better than mine, but that's entirely beyond the point. So this is the situation. And what about experiments? The most puzzling and difficult to understand experiments are reported by Thomas Gregor and his group last year. And what they did is they were able to consider two parts on the chromatin by very clever genetic manipulations. They were placing promoters and enhancers of particular genes in different parts of the genome, varying the distance, the genomic distance between them. And by doing this, you see it's part of B part of figure 1. This is D part of figure 2. This is A part of figure 3, I think. So it's huge information in this article, but I collect only a few pieces. So here is the distance between these two units. And here is the distance. Pay no attention to this. It's unrelated to us now. So this is the distance. Well, it goes at large distances, it deviates, but here it seems to follow beautifully this s to the 1/3 power. So someone like me should say, oh, triumph, it's really proven. This is a crumpled model. I don't want to read this paper anymore. I want to stop at this point, but I read further. So now you consider mean square displacement to one of these guys, and it goes like t to the 1/2. And now you consider the distance between these two guys, which is sort of a pair correlation function, and you again see that this goes like this. So I remind to you, measured subdiffusion exponent is 0.26 here. Well, it's half of this, it's quarter, so basically 0.26. Classical rows for without any secrets, Gaussian chain, 0.25. Excellent agreement. And now all this high-level, beautiful, powerful theories, complete dramatic failure. So that's where we are. That's the state of affairs here. It's complete puzzle. To tell the truth, I suspect that the key is perhaps here, that this perhaps is not quite crumpled, but something else. So, but I don't know. This is a puzzle. And I think, if I remember correctly, this is my last slide. Well, I am prepared to talk to about high dynamic screening, but that probably should be done next time. So, thank you, and I am prepared to try to answer any questions that you have.